Related to autism, sometimes I'm wondering whether I myself have some, because I'm very interested in research and then get addicted. But then my wife told me I couldn't manage my finance. So then I tried to learn that. So then I become addicted. You know, someday you play with the stock market, then you lose money. So this kind of thing, it's kind of, sometimes we have to control our behavior, I guess. For today's talk, I want different slides, some slides for the patient family, and some slides for scientists we have here. So I give a different title here. So this title is inspired by the famous book by Charles Darwin's Origin of Species, and here I'm wondering what's the origin of disease. One key cause is mutation. So in genetic disease, in cancer, even during COVID, for example, the virus evolution is because of mutation. So in here, let me see. Here you have the lysine, this is K. So if that changes to a stop codon, as stop, then the protein truncated. So you just need one nucleotide change. So in this case, you just need to the AAA to TAA, just one. This is, when we have a text, language, we copy from, let's say, English from one to another. We make mistakes in the cell. The genetic information needs to be copied. And during that copy process, there is something called replication errors. So that is the main source of mutation. And this is random. First place, it's readily random. So that's why, sometimes I met patients' parents, and they ask me, how could the kid get a mutation? It's random. If it's de novo, random. Even if it comes from parents, the initial is also random. So that's why try to understand this is it happens by chance. Okay, with that. So I studied the research. This was many years ago when I was a postdoc. At that time, I was at NIH. So there was these two papers about this different mutation. Chromosome translocation in leukemia. So one is MOS, now known as CAT6A. And this was a published group in Boston here. And another paper, a similar paper on a different, from another lab from UCSD about related protein. So here you can see the translocation between MOS and different, this is also acid transferase. So that causes leukemia. So this is a somatic mutation. It's not the whole body, only in certain blood cells. So that causes leukemia. So at that time, I tried to look for, I had a position at McGill and tried to find something to do. And one thing we wanted to test whether this was really acid transferase. And that turned out to be true. And we also identified a related protein done by a former postdoc and two graduate students. And this we call the MORPH, now known as CAT6P. So this is the molecular level. So to demonstrate that the protein is an acid transferase. So then afterwards, I think Dr. Wolf and Thomas' lab, I think they were still in Germany at that time, I guess. I demonstrated that this protein is important for brain development. And then other lab just show that both genes can be rearranged in leukemia. And chemical A, so this is lysine residue, just add this ester group. That's what we call the estylation. So we all drink alcohol. This is the part structure of the alcohol, ester group. Now we know that in addition to this ester lysine acetylation, there is called propionylation, and some others. So this relate to short chain fatty acids that Dr. Kelly asked. And so then those proteins, they do not work alone. They work with several other proteins, especially this protein called BRPF. This actually can activate those enzymes. So they are very tight. It's like in a company, you have a CEO, you have different supporting people. And so this is activates those enzymes. So without those proteins, the enzymes activity is very low, even in vitro. And this was done in a collaboration with Dr. Cote at the Laval University. And from here you can see that this is the catalytic domain of the enzyme, so that mediated the interaction of the BRPF protein. So we're wondering what's the function of this BRPF1, and a former student did a knockout of the mice knockout. As you can see here, this is like at the middle of embryo development. This is a normal embryo, and this is the homozygous knockout. You can see the development. So the protein the gene is very important, especially in the head regions. It's completely missing. So if only developed in the blood system, so that's called bone marrow failure. So this is 12 days after birth, and at 5 days is still okay, but afterwards the bone marrow just has problems. And if knockout only in the cerebellum, so the hippocampus is affected. So this is important for memory and learning and intelligence as well. You can see this is 10 days and 12 days. This is a control, and this is the CA region, and this is dandy gyrus. You can see that this is it's really affected. So at that point, we want to ask whether there are any human mutations in this gene. And we collaborate with Dr. Philippe Capo at the University of Montreal and other clinicians in US, Israel, and also in Europe. And we identified multiple patients, and I should say this is only one copy of the gene is affected, and it's de novo. And the main problem is intellectual disabilities. There are some other features as well. I should mention in mice, heterozygous, we didn't really see major problems. And those mutations, some of them truncation, some of missense mutation. And this just affects the ability of this protein to activate CAT6A and CAT6B. So basically CAT6A and B get instruction from this protein to acetylate histones. And this is a complicated slide, but maybe related to treatment. Over here you can see that in cells with different histone deacetylase inhibitor, and this is a beta hydroxybutyrate and sodium butyrate. You can see the acetylation. Not just acetylation, there's another function modification called propionylation, and this is increased. And in patient cells, that this treatment or this propionate also enhance the acetylation. Not only that, also rescues the defect. So basically in cell you have the acetylate and propionate, and then affect the acetyl-CoA and the protein-CoA, and then affect the acetylation and propionylation. So this is striking because usually for enzyme we have, usually they use like a one co-substrate, in this case a different co-substrate. I guess, for enzyme, maybe this for the enzyme to function in different conditions, so become more versatile. I guess that's the reason. So At that point, the question we asked was, because this is a family of enzymes, so a lot divided into 3 groups. This is the last group called this family, contains Cat5, 6A, 6B, Cat7, and Cat8. The question, obvious question, so whether any mutation in those other genes. And for the rest of the talk, I will focus on this one, Cat8. So basically, we did a knockout, and actually the phenotype is even more striking in mice. And this is a control, and 5 days after birth, and this is cerebellum. You can almost totally missing. And this is just a different stage, and I think at 16.5 you see some, but later you see major defects. So this is the neural stem cell, neurosphere, the neural stem cell wild type, and this is from knockout. So there's a neural stem cell defect. So that's probably something we should also look at in Cat6A and Cat6B mice. And then also the question whether there are patients with Cat8 mutation. So those we found collaborated with different clinicians, including Dr. Kapoor. We found multiple patients. All of them have a single copy affected, except this patient from Israel. So she has both copy affected. One of copy actually has a truncation mutation. So that's still mystery to us, why this patient has both alleles affected and could still survive. So it's quite puzzling to me. And that other mutation is also very important. Not just a catalytic domain, so there's this other domain that may be involved in binding to histone or chromatin. So normally, basically, you have, during normal development, you have Cat6A and regulate histone acetylation. In this case, the site is different from Cat6A and Cat6B. And then regulate the neural stem cell or progenitor and regulate the brain development. And in patient, if this is affected, it then cause problems. So the last question is what happened to the remaining family, Cat5, Cat7. Cat5, this was done through a collaboration, and that affect mainly the cerebellum and the sleeping and epilepsy. I don't think they show intellectual disability, at least to my knowledge. So then the question, what's about the Cat7? So that is still open question. At last, a few slides, just more general. So then I mentioned the origin of mutation. So those results provide molecular diagnosis, then related to therapeutic and innovation. So I guess, it's more like a reproductive decision. So let's say for a young couple of parents, so if one kid has a mutation, so it's a good idea to have the second kid. So because the mutation is random, unless the father has a lot of mutation in the sperms, I think it's a good idea. So then therapeutics, sometimes we all try, but this is developmental problems. Maybe we will see. And I attended this, and also 4 years ago, when I see the patient's family and the patients, sometimes I'm really moved, and so truly it's inspiration to me, and give me good ideas. I need to work harder. And this is through collaboration with Dr. Kapoor and Kote and many other researchers and former trainees. And I just mentioned that this one now has a new disease name, named by the first three authors of the paper. I will take some questions if you have any.